Hello, tonight we're going to talk about mitosis. The cell cycle is basically the life of the cell condensed into a circle, a pie graph, to represent the various stages of the cycle. Most of the cell cycle is spent in interphase. Interphase is divided into three parts, G1, S, and G2. And then you have the meiotic phase, which is mitosis, followed by cytokinesis. During G1, the cell is going to grow and go through its normal cell activities. Most of the cell growth for the cell is going to happen during this phase. Then the cell enters the S phase, which stands for synthesis. This is when DNA replication occurs. So in a human, you go from 46 chromosomes to 92 chromosomes. After that, you enter the G2 phase. During the G2 phase, you have a little bit more cell growth, but not a whole lot. But you continue with normal cell activities. Then you enter mitosis. During mitosis, the chromosomes are going to separate into two nuclei. And then you enter cytokinesis, where the cytoplasm divides and you divide into two cells. Some of the terminology you need to know. A genome is all the genetic information in the cell. Chromosomes are the DNA when it's compacted into a small organized state. Somatic cell is a normal cell in a body. A gamete is a reproductive cell in the body. So in you, most of your cells are somatic cells. However, the gametes would be eggs or sperm. Chromatin is DNA when it's all unwound. Now there's a little terminology thing here that I find a little annoying. I wish they didn't do this, but I have to explain it to you. When you are in the G1 phase, or whatever, you have one piece of DNA and it's called a chromosome. Okay? When you go into S, it's replicated, it's duplicated, and you have two of them. The two of them together are called a chromosome. Each one is now called a sister chromatid. So they're using the term chromosome for a single piece of DNA or for two pieces of DNA connected at a centromere. I hate that they do that. But then the sister chromatids divide, so the chromosome divides. Each one becomes, get it, a chromosome. So they're using the term chromosome to mean two completely different things. And I hate that they do that, but they do. Sorry. One thing that you probably have never been taught before, so this is a new thing. Where the spindle fiber connects to the centromere region, there are a bunch of proteins there that allow this connection to occur. Those proteins are called a kinetochore. Okay? That's just kind of fun. New terminology, new things to learn. Okay, so let's go through interphase. We're going to go through all the phases of mitosis now. Afterwards, we're going to talk about some of the difference between plants and animals in mitosis, and then we're going to look at how mitosis is regulated. Okay, so we go into interphase, G1, S, G2. So the cell's going to grow, the DNA is going to replicate, a lot of the organelles are going to duplicate, go through more growth, and then we're going to start into mitosis. One thing that does happen is the centrioles, a pair of centrioles is called a centrosome. The centrioles are going to replicate also. So now you've got two pairs instead of one pair in a cell. So here are your two pairs of centrioles called a centrosome. So many C words in this stuff. Okay, prophase. During prophase, the prophase is actually kind of two phases together. There's prophase and then there's metaprophase. During prophase, the chromosomes are going to condense. If you remember when we talked about DNA, what happens is that the DNA winds around little protein molecules called histones. And they wind around and around, and the histones collect and gather together, making the chromosome. They do that because if you have a whole pile of DNA, chromatin, unwound DNA, it's very hard to move it around the cell, especially if you've got 92 of those moving around a cell. Excuse me. Well, by condensing it into a chromosome, it's much more easy to move the DNA around the cell. Okay, so the DNA condenses into chromosomes. The nucleolus is going to break down, and then you enter pro-metaphase. During that time, The nuclear membrane breaks down and the meiotic spindle forms and connects to the chromosomes at the kinetochore. Okay, so there's a picture of prophase. The DNA is still chromatin. It's starting to condense, but it's not fully condensed yet. This is prophase. This is prometaphase, where you've got the chromosomes, it's connected at the kinetochore, the nuclear envelope is breaking down. Okay. So I just mentioned the centrosome and the kinetochore at the middle. That's where the microtubules connect. During prometaphase, the microtubules are going to play a little bit of a tug of war back and forth, trying to align the chromosomes at the metaphase plate. As soon as they are all aligned on the metaphase plate, it's called metaphase. After metaphase, then here's the metaphase aligned, then we enter anaphase. During anaphase, the chromosomes will separate. The two sister chromatomes will move opposite directions in the cell, and you will have chromosomes again. I know it's the terminology, but that's what happens. Okay, so the sister chromatids are moving opposite sides. It's now called a chromosome again. Going opposite sides. That takes you to telophase. During telophase, the opposite of prophase is going to happen. The nuclear envelope reforms, the chromosomes uncondense back into chromatin, the kinetochores break down, the microtubules break down, all of that. And then we start into cytokinesis. Now, there's a difference here between cytokinesis in plants and animals. In animal cells, cytokinesis will form a cleavage furrow, which is on the outside and it pinches off towards the middle. In plant cells, it starts with a cell plate in the middle of the plant and it grows out to the outside of the cell, dividing the cell. So how cytokinesis occurs in the two cells is different. This is an animal cell, so it's pinching in, forming a cleavage furrow. Now, one other difference between plant and animal cells before we watch this video is that plant cells don't have centrioles. Animal cells do. Both have the microtubules and the spindle, but what they don't have is that centriole that it connects to. It's just kind of out there in outer space. Well, or cytoplasm, wherever you want to talk about it. Okay, let's watch this video.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands, called microtubules, appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of 2 cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the 2 halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the 2 new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The 2 new cells are now ready to grow and perform their specialized functions.
Let's look at how mitosis is different in certain organisms now. Bacteria, because they don't have a nucleus, don't go through mitosis. What a bacteria does is they first replicate the DNA. They pry open the DNA at an origin. Once it's origin of replication, they pry it open, they replicate it. Their DNA is a circle, so they're just going to copy in both directions. Zoop, 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 zoop. Okay? And then once you have 2 copies of it, the cell goes through binary fission and divides. And that's the end for a bacteria cell. But eukaryotic cells do things a little bit different. Okay, here's a bacteria. Some unicellular protists, like dinoflagellates, do things a little different. First of all, they keep their nuclear envelope intact, so it doesn't break down. But if you look on a picture, you'll see the microtubules go through the nuclear envelope, through kind of like a tunnel, all the way through. The DNA is actually connected to the nuclear envelope. The microtubules pull the nuclear envelope in half, pull it apart, and thereby separating the chromosomes. Kind of different, but kind of cool. The next big change occurs in another unicellular protist called diatoms. This time they still keep their nuclear envelope intact, but the microtubules are now on the inside of the nuclear envelope, and they are connected to the nuclear envelope membrane. And what they do is each one is connected to a chromosome. Instead of having the chromosomes connected to the nuclear envelope, they're free-floating. This connects to them. And then the microtubules also push apart and separate the nucleus. And then you get to plants and animals, which we've just gone into detail about how the nuclear envelope breaks down. Your centrioles connect with a microtubule to the kinetochore, pulling the chromosomes opposite directions. Now don't forget though, plants don't have the centriole. They don't have the pair, the centrosomes. Let's regulate the mitosis. Okay, there are 3 checkpoints in mitosis. There's a G1 checkpoint, a G2 checkpoint, and an M checkpoint. The first one is really not understood very, very well, but at the G1 checkpoint, one of 2 things is going to happen. Either the cell is going to continue through the cell cycle and divide, or it enters a different phase called the G0 phase. During a G0 phase, the cell can grow, but it never replicates. You find this in nerve cells and muscle cells. You know, muscle cells can get bigger, but they never divide. You don't get more muscle cells in your body. Okay? Nerve cells, however, do have the capability of regenerating. When a nerve has been damaged, it can be forced out of G0 back into G1 and then replicate. Muscle cells can't do that. So while the G1 is not really understood very well how it does this, they know there's some proteins involved called cyclin-dependent kinases, that's CDKs, some cyclin molecules. Again, it's not really understood, but the G2, which uses the same molecules, is understood pretty well. So I'm going to talk about the G2 checkpoint, knowing that the G1 is probably very similar. There's also the M checkpoint we'll hit right at the end. Okay, so this is what happens. In the cell, at the end of mitosis, you have this molecule called cyclin-dependent kinases, which are there. They're inactive. They're just hanging out there. They don't break down. They're there the whole time the cell's alive. You have another molecule called cyclin. Cyclin is built. As the cell ages, it builds more and more cyclin. Cyclin connects to the CDKs, forming another molecule called a mitosis promoting factor, MPF, mitosis promoting factor. What that does is it says, hey, I've got enough of this mitosis promoting factor, let's go into mitosis now. So that's what it tells it to do. During mitosis, the cyclin is broken down, so at the end of mitosis, the cell starts back over with no cyclin. And so then it can build it up again and tells it when to divide. So here's what happens. At the end of mitosis, you have very little cyclin that builds up throughout G1, S, G2. When you get a whole bunch of it, it connects to the CDKs, which make that mitosis promoting factor. You get a bunch of that, and that says, oh, enter mitosis. And then you go through mitosis. Cyclin breaks down, which means the MPF is going to break down. And so you start back over with nothing. Now, I like this slide better than that slide. This is my favorite slide in the whole PowerPoint. At the end of mitosis, you have the CDKs. CDKs are here the whole time. Okay, they're here the whole time. There isn't any cyclin. So we're going to start building cyclin up here in this outer circle. Once you have enough cyclin, see the shape? Cyclin connects to CDK, creating the mitosis promoting factor. When you have enough of that, the cell says, oh, let's enter mitosis, and so it does. Then the cyclin's degraded, So you lose that part, so you just have the CDK again, and then you just start back over. Now, the M checkpoint, it occurs during metaphase. During metaphase, during the M checkpoint, it waits until all the chromosomes are lined up on the metaphase plate. It won't enter anaphase until everything is lined up. As soon as they're lined up, then it says, okay, go ahead, go into anaphase. And then the chromosomes are separated, you go through cytokinesis, the cells divide, and each cell supposedly has the same number of chromosomes. That's what the M checkpoint is for. There are a few factors that can affect the cells in the body and also in cell culture. So if I want to grow some like cancer cells in the lab, or I want to grow some muscle cells or nerve cells in the lab, there's some things that can affect the growth. Okay, now they affect in the lab, but they also affect inside the body. First of all, you have to have growth factors which promote cell division. So you have to have growth factor. The second thing is density. If the cells are too dense, they will not divide. No matter how much growth factor you give them, they will not divide if there isn't space to divide into. Also, if there isn't any place to anchor, the cells will not divide. You have to have an anchor, an anchorage spot. However, cancer cells will divide without any of those things stopping them. They're just uncontrolled cell division. And unfortunately, that's what cancer is, uncontrolled cell division. Okay, that's it for tonight. Your homework tonight, I would like you to draw the 4 stages of mitosis: prophase, metaphase, anaphase, and telophase, and describe what occurs in each of the 4 phases. Okay, just a nice little drawing. I'll see you tomorrow. Bye.